In subsequent studies, it was proven that the injection of an mRNA strand of a particular virus will cause the body to synthesize the relevant proteins in the process of translation during protein synthesis. The proteins will then be recognized as foreign, therefore allowing the body to create antibodies against the targeted virus, consequently allowing for specific and long-lasting immunity. Jonty will further explain how scientists today utilize the concept of protein synthesis to create mRNA vaccines that allow for immunity against particular viruses such as COVID-19.
So what about the biological mechanism of mRNA vaccines? When a virus enters the body, it attaches to our cells and injects its RNA, telling our cells to produce more of the virus. However, these viruses are recognized as foreign and attacked by our body's immune system. They produce antibodies which attach to the virus and stop them from sharing their RNA and marking them for destruction by the body. This can take days, however, and often produce intense, harmful symptoms in the process. In terms of the creation of an mRNA vaccine, most commonly, a sequence corresponding to a respective mRNA is inserted into a plasmid. These plasmids can replicate themselves in controlled conditions, and an enzymatic reaction triggers the mRNA synthesis. The synthesized mRNA is purified and encapsulated in a lipid nanoparticle, which allows the mRNA to reach the cells in the human body, as the body's immune system quite easily destroys the mRNA. More common and traditional vaccines rather see weakened or dead parts of pathogens injected into an individual. The outcome is the same. It triggers an immune response in the body, developing the required T cells and antibodies to kill off the pathogen. Using COVID-19 mRNA vaccines as an example, these vaccines must first reach the cytosol, found within the cells, to translate antigens for an immune response. DNA vaccines, in comparison, must cross the nuclear envelope, whereas RNA vaccines only must cross the plasma membrane. The mRNA in these vaccines contain instructions for our cells to produce an isolated spike protein. These instructions interact with ribosomes in our cells to produce these spikes rather than the whole virus itself. As they move through the ribosome, codons in the mRNA translate for specific amino acids, added in by tRNA. When completed, the polypeptide, the desired spike protein, is released from the ribosome.
Created using Powtoon.